ended up figuring out is like, as a person, super nice. As a professor, I probably didn't want to take the class. All right, let's see where we get today. For your exam on Monday, remember after class today, we're going to go up and look at some of your results. The other option, if you can't come today, you can come Monday after class, but that's about it. So make sure that you come up today. We'll have the reagents to do that and record our results. So we are in the section on miscellaneous gram-negative rods. We talked about Francisella at the end of Wednesday, and we're now in a genus called Legionella. So Legionella is generally a respiratory pathogen. Of course, they're gram-negative, right? We're in that gram-negative group. They're aerobic, and they have fastidious growth requirements, so they're not just going to grow on anything. You have to sometimes, give them a little bit more nutrition, either through like blood agar or something like that. They are facultative intracellular, so they can replicate outside of cells, but they also replicate inside of cells. And in the wild, right, or not even the wild, just like outside of the human body, they're often found in amoeba. They're like, not like a symbiont almost sometimes of amoeba. They, in the host, like us, will go into alveolar macrophages, monocytes, epithelial cells. But again, they're mostly respiratory, so they're going to be hanging out in the respiratory tract. And they can evade destruction by those immune cells, the macrophages and the monocytes, by preventing phagolysosome fusion. And they can kill host cells. They have a lot of different enzymes that are proteolytic. They destroy proteins to kill host cells. So they can cause some major issues in the lungs. And they are only killed if macrophages are activated through the T helper 1 pathway. Otherwise, they can evade macrophage destruction. So a lot of the other genera, Bartonella, Bordetella, Francisella, Listeria, right? They're all named after people, Escherichia. This is named after a group of old men, the Legionnaires, the American Legion. So it's a social organization. It's, again, mostly old men. So it causes a disease that we call Legionnaire's disease. And we call it that because the first time it was ever characterized was it caused an outbreak of disease during an American Legion convention at a hotel in Philadelphia. Depending on the immune status, the mortality rate can really vary. And again, because we saw this a lot in old people who don't have the best immune system, right? They are more susceptible to more dangerous disease. Generally, we see a 2 to 10 day incubation period, followed by fever, chills, dry cough, so not a mucus producing cough, then headache, and it can go multi-organ, but primarily the primary disease it will cause is pneumonia. It is a respiratory, it's an inhaled pathogen. So it was ID'd for the very first time in people in 1976 in Pennsylvania. There were 200 cases and approximately 30 deaths at the Legionnaire's convention. So that's why it's Legionella, Legionnaire's disease. In Flint, Michigan, they were having all of those issues with their water supply being contaminated. They were also seeing cases of Legionnaire's disease. And so where we primarily see this is through the air conditioning system, so through the HVAC. The condensation in the air conditioning promotes the growth of the amoeba that carry the bacterium. And then when you turn on the AC and you blast that into the room or into the hotel, or as we'll see, into the hospital, patients can be exposed and develop the infection. So this is an outbreak of Legionnaire's disease in England. In 2003, an elderly man, he wasn't part of the American Legion because he was in England, but still, an old guy, died of Legionnaire's disease. 3 days later, an elderly woman died of it as part of a surveillance. They found at least 2 additional patients. Further investigation revealed 28 linked patients with onset of disease between October and November. Positive urine antigen tests. 4 had high antibody titers. 2 were culture positive. The implicated source of the outbreak was a cooling tower that had recently been restarted after a period of inactivity. So the condensation from there. After the tower was closed and recleaned, the epidemic was terminated. This outbreak illustrates the difficulty of recognizing the problem when the infected individuals may present to different hospitals. This is particularly a problem if the source is located in a hotel or vacation area. So imagine, right, a 2 to 10 day incubation period, and let's say, like I stayed in a hotel last weekend. So I was in a hotel last weekend, and now I start to feel sick. Well, I was in Santa Cruz. So I'm no longer in Santa Cruz. So I would go to the hospital here in Turlock, or I would probably go to Modesto because that's what my insurance covers. So I'd go to Modesto to the hospital. But there were people from Northern California. They might go to a hospital in Sacramento or Chico or Southern California, or from another state or another country. It's hard to figure out that we all were actually exposed at a hotel in Santa Cruz until all of these people are interviewed. And then all of us remember where we were likely exposed. We're all diagnosed correctly with the appropriate infection. And then every single one of those public health agencies says, hey, I have a patient who stayed at a hotel in Santa Cruz the night of the 23rd and has Legionnaire's disease. And then the person from Sacramento calls, and the person from Washington calls, and the person from LA calls, and the person from England calls. So it can be really hard to track these things. And now people are staying the 24th, 25th, 26th, 27th, 28th, 29th, being exposed. So with something like that, any kind of disease, not just Legionnaire's disease, any kind of disease that comes from somewhere like a hotel, it can be really tricky to track back where it came from. So it can be diagnosed through serum antigens and nucleic acid tests. Patients will mount an immune response. It can be cultured. It's just a little bit trickier. You might want to use something like blood agar. We don't see a ton of antibiotic resistance, but so we can use different antibiotics to treat it, macrolides and fluoroquinolones. You just want to make sure that because this microorganism can go intracellular, that you choose antibiotics that are also capable of going intracellular. Not every antibiotic is good at getting into host cells, so you want to make sure you get an antibiotic that can get into the host cell and potentially target that microorganism that's in there. So in order to make sure you get rid of the organism, you would want to either hyperchlorinate or superheat the water supply. You can use copper-silver ionization. A lot of times metals are really good at getting rid of bacteria. That's why even in some hospital rooms, the hospital beds, they're starting to coat the handrails with things like copper because it has antimicrobial properties. So it's not an uncommon infection. This is MTA, so it's like the transportation system, like the subway in New York City. Legionnaire's sickens 9 people at the Graceland Hotel. Going to pay your respects to Elvis. And then this was the hospital where I used to work. BAMC employees test positive for bacteria that causes Legionnaire's disease. And then Legionella detected at the Royal Children's Hospital. So it gets into these, oftentimes air conditioning systems is where we're going to see a lot of that. And when it happens in a hospital, that can be very, very dangerous. Yes?
Don't most hospitals have like some sort of human air filtration?
No.
So if there is, would that prevent it?
If they had all of the outflows, with a HEPA filter, yes. So I shouldn't say that hospitals don't have HEPA now. Because of COVID, most of them really started upgrading their systems, but they used to not. And so it was relatively common Like the last person that actually died of smallpox died because somebody was working with it in a hospital like 2 or 3 floors away, and it came through the air system and it killed somebody. So they didn't filter their air like they should. But with COVID, I know more hospitals have started doing that.
So even if like the reservoir system is like contaminated with Legionella, every outflow is like.
Well, so the thing is it can be like in the ductwork leading towards the outflow. So if there's any condensation in that ductwork, that's where it could be. And so if you're not filtering right before it comes into the room, even if you filter like the main air supply, that area could still be contaminated. So you'd have to have all of the ducts locked basically with filters. Yeah, that's the challenge. So you could, if you had at least the, centralized HVAC system with some sort of like filtration, you could probably prevent it from spreading throughout the entire facility. But if it's in one particular area of the ductwork, it might still spread within that area. Okay. Oh, and then 73 cases in Metro Detroit, so a lot. So that's where you have to be cognizant of what's going on in the AC. It's going to make you sick. And then I added this one because I don't know if you've ever heard, like, people will tell you, oh, dogs' mouths are, sterile, and if they lick you, it's totally fine. Like, okay, first of all, it's not. Dogs' mouths are gross. Everybody's mouth is gross. Everybody's mouth is contaminated. I think we all know that. We've done the experiment. This bacteria, Capnocytophagea canimorsus, is a commensal bacterium that's often found in the mouths of dogs and cats. And what can happen is if, just like we saw with the Bartonella Hensley, if your cat licks you or your dog licks you and you have a wound, you can get infected. And so symptoms can appear anywhere from 2 to 28 days after initial exposure. Fever, vomiting, diarrhea, rashes. It causes that disseminated intravascular coagulation. It can cause meningitis. And so after, a lick, or more commonly a bite, the chance of infection after a dog bite is anywhere from 3 to 20%. After a cat bite, up to 50%. And you can grow it on blood agar or chocolate agar. So if you have a patient who's been bitten by a dog or a cat, I know a lot of the times what we worry about the most is something like rabies, because biting is a symptom of aggression and aggression is a symptom of rabies. Bacterial infections can also be relatively common after an animal bite because they have a lot of microbes in their mouth. And when they bite, especially when it breaks the skin and draws blood, those bacteria from their mouth, their saliva, their teeth can get directly inoculated into the wound. Okay? And so this is one that can be really dangerous. So you may want to consider, if you're a physician with a patient with a dog bite, a prophylactic type of antibiotics to make sure that they don't develop an infection of the wound. It's relatively treatable. Not a lot of drug resistance. It's not a common bacterium. Most of you have probably never heard of it before. But you may have read headlines like this. A man's legs were amputated after dog licks lead to infection. So what happens with that DIC, that disseminated intravascular coagulation, a lot of times with any bacterial infection that can cause that type of septic infection, when you have those blood clots, they tend to get stuck in the capillaries in the extremities, and they cut off blood flow to the extremities. And so that can cause tissue death, right, necrosis, and additional infection. And so oftentimes the only way to treat that is amputation. So you will see relatively commonly patients who have infections, they'll report lose their toes, lose their fingers, lose their feet, lose their limbs. Okay? Because that's where you're going to have a lot of the necrosis you're going to have really significant issues there. And then this one, a Wisconsin woman's death was linked to bacteria from dog saliva. So not so common that you'll have heard about the bacterium, but common enough that you've probably heard about it in the news, at least in terms of headlines like this, and other bacterial infections from dog and cat bites. So when you have a patient with an animal bite, again, you may want to consider not just worrying about rabies, but also do some prophylactic antibiotic therapy. All right, questions about miscellaneous gram-negative rods. Okay. Let me make sure I put these slides up. Oh, I did. Good. Okay. So our next section is Clostridium, and that's probably as far as we're going to get today. We may not even get all the way through Clostridium, and that's fine. So Clostridium is, we're jumping back into gram-positives. Clostridium is gram-positive. It is also spore-forming. Not all species of Clostridium form spores, but many species of Clostridium can. Gram-positive, and you see here I put like most of the time. It's not that they don't have the right organization. It's just that sometimes when you do the staining, just because of the different properties of every individual species, they may not always look as deep purple as you expect from a gram stain. So, but they are organized like a gram-negative bacterium. They have the thicker layer of peptidoglycan. They have the single membrane, right? Peptidoglycan membrane. And they can form spores. So that's what's pretty important. The majority of the clostridial species, especially those that cause human disease, are actually obligate anaerobes. So the other genus that we talked about that forms spores is Bacillus, and Bacillus is aerobic. But the major species that cause human disease from Clostridium are anaerobic. Okay. So they are obligate anaerobes. Clostridial species are ubiquitous. They are found everywhere. Soil, water, sewage, so they can be found in the GI tract. And the majority of species are harmless. But there are a handful that we talk about that cause human disease. There are 4 in this section. Like I said, I don't know how many we'll get through today. Whatever we get through today is what we get through. And the rest later. But the species that do cause human disease cause a variety of different types of disease. So we see some that can cause food poisoning and gastrointestinal illness, tetanus, which is a spastic paralysis, meaning the muscles are constantly spasming. Botulism, which is the exact opposite, it's a flaccid paralysis, the muscles no longer contract. And even gas gangrene, so deep soft tissue infections. So again, there are 4 pathogenic species in this section that we're going to talk about. These are other ones that cause disease. So most commonly, if you see a clostridial infection, you're going to be seeing Clostridium difficile, which they'll sometimes call C. diff, or Clostridium perfringens. But of course, we also do have Clostridium botulinum and Clostridium tetani as, uncommon but not rare. So there are other species that can cause similar types of infection, but these are the 4 big ones: difficile, perfringens, botulinum, and tetani. So Clostridium difficile has gone through some, taxonomic reorganization. I don't know what they're doing half the time. They change the names all the time. So you'll find some papers that will call it Clostridium difficile. You'll find other papers that will call it Clostridioides difficile. I don't care. Just call it C. difficile. It makes it a lot easier for you. Both names mean the same organism. They do form spores. They grow relatively rapidly. I have seen some papers talk about that they can replicate every 8 minutes. So they grow rapidly, but they will die when exposed to oxygen. That's what happens to obligate anaerobes, right? Obligate anaerobes don't do well with oxygen. They don't tolerate it. This particular species of Clostridium is the one that's really highly associated with causing gastrointestinal disease. And the bacterium produces volatile fatty acids. And I don't have personal experience with this, but they say that when a patient has diarrhea caused by C. diff, that the feces produce a barnyard smell, that it smells more like manure than you would think that human feces would smell like. And I know it's like, oh, now we're talking about the way poop smells too, but sometimes that can be super informative. Like, we all know that feces doesn't smell good, but when it smells more like cow poop than human poop, that could be indicative of a C. diff infection. Because a lot of the reason that manure smells like manure is because of the volatile fatty acids produced in the GI tract of those animals. So it has a barnyard smell, they say. Again, I've not had the infection and I've not smelled the infection, so I can't say for my own personal experience, but that's what they say. Most commonly with C. diff, what we see is in our hospitalized patients who have taken antibiotics for something else. When they take those antibiotics and it alters the normal microbiome of the GI tract, that opens up the GI tract for infection by C. diff. So this tends to be a hospital-acquired infection. It goes around the hospitals. Remember, it forms spores. Spores are pretty resilient to drying, to cleaners, and so it can go around the hospital pretty easily. In the majority of patients, it can be self-limited, right? You have diarrhea, and eventually, you can clear it. But in some patients, for whatever reason, the reason people experience disease different, right? We're genetically different. We have different microbiomes. In some patients, it can be more prolonged. And in some patients, it can cause more severe disease than just diarrhea. It can cause, they call it pseudomembranous colitis, but basically you have plaques forming on the walls of the colon. You have tissue destruction occurring, profuse diarrhea, abdominal cramping, fever. So the bacterium mostly causes disease through the presence of a couple of different toxins. There's toxin A and toxin B. Toxin A is a classic enterotoxin, and the way that it works is, if you picture your intestines, you've got all of these cells, right, all right next to each other, any tissue, right? You have all the cells right next to each other, and that prevents fluid from leaking in and out. But this particular enterotoxin can disrupt those tight junctions, and that allows fluid to get into the intestines. The toxin B is a cytotoxin that destroys cells, it kills cells. And strains that do not have toxin A can still cause disease. So toxin B alone is enough to cause disease. And what you see in this image here, these are some of those plaques that you get with that pseudomembranous colitis. More recently, scientists have identified what they call a hypervirulent strain, and it produces a third toxin. They call it binary toxin, and it actually leads to some more tissue destruction. But in this particular strain, it also expresses the other two toxins more highly. So it upregulates the expression, or has upregulated expression of the enterotoxin and the cytotoxin, and it makes a third toxin. And it's resistant to fluoroquinolones. So I guess if you had your choice of which strain of C. diff would you want, that should be last on your list. So it's drug resistant, it has upregulation of the two toxins, and it has the presence of a third. This bacterium is part of the normal microbiome in some people, not a lot. As long as it's not able to take over, we can coexist. And that's how we coexist with a lot of organisms in our GI tract that are technically pathogens. Generally, though, when you have antibiotic therapy, it allows it to take over. And that's when you have the problem, when you've wiped out the competitors and you allow it to take over. And so to diagnose the infection, you generally can look for the bacteria or the spores, right, in the feces. You can culture, but you have to be careful. So you have a patient who has diarrhea. Is it bacterial? Is it viral? You think it's bacterial, okay, but is it Enterobacteriaceae? Is it Bacillus cereus toxin production? Is it staph toxin production? Is it Vibrio? What's causing it? Is it Campylobacter? Is it Helicobacter? Is it Clostridium? All of those things have to be cultured in different ways. So if you suspect Clostridium, you would have to culture it under anaerobic conditions. Okay. You can look for the presence of the toxins through serology, but you can also diagnose it based on symptoms. If you have a patient who came into the hospital for any other condition and did not have diarrhea, but was given antibiotics for something else and developed diarrhea, Clostridium might be one of your top things to look for. You can do, again, the nucleic acid testing to look for the genes that lead to toxin production. Sometimes if you discontinue the original antibiotic, the one that's killing the normal microbiome, that can help because the normal microbiome can regrow. Metronidazole and vancomycin are therapies that are generally pretty good at getting rid of Clostridium difficile. We will see metronidazole again. Metronidazole is one of those weird things it's not that it is an antibiotic that targets bacteria, but it works really well at killing anaerobic organisms. So we'll see it as a treatment for one of our anaerobic parasites. And then sometimes you have to do 2 courses of antibiotics because there could be spores left over from the first infection. And if those spores germinate, you have a second infection. But generally, after 2 courses, the patient is okay. But if you have a patient who just for whatever reason is not able to clear the infection, there's a really great treatment. Repopulation, or a fecal microbiome transplant. That's the more scientific way to say it. And it's almost exactly what it sounds like, a fecal microbiome transplant. You take fecal material from someone who is healthy, you remove as much of the solids as possible to try to isolate the microbial community, and you give that microbial community as a treatment to your patient. It can be done through a suppository. It can be done through like a nasogastric tube, or through a capsule that the patient can swallow. It sounds gross. In theory, it kind of is. You're getting somebody else's fecal bacteria, but it works really well. Clostridium, for whatever reason, if you put other bacteria in there, the Clostridium's like, whoa, I can't deal with this. And it tends to cure the infection. So giving a patient a healthy microbiome through one of those mechanisms can usually help them kick the Clostridial infection. They do this. I talked to a pediatric oncologist from Valley Children's Hospital once, and the kids get C. diff infections all the time, the kids that have cancer, and this is what they do for them. I mean, the kids are already suffering so much, and now they have all this like nasty diarrhea. This like really helps to clear it up. So it's a relatively common thing to have happen in the hospitals. So this is a classic presentation of C. diff disease in a 60-year-old man who got a transplanted liver 5 years ago. He showed up to the hospital because he had crampy abdominal pain and severe diarrhea. 3 weeks before admission to the hospital, he got a 10-day course of trimethoprim sulfamethoxyl because he had a sinus infection. On physical exam, the patient had a fever, had moderate abdominal pain. A CT scan showed thickening in the colon, but no abscesses. A colonoscopy, so the colonoscopy is when they put the camera up, showed numerous whitish plaques and kind of redness in the mucosa consistent with pseudomembranous colitis. Empirical theory with oral metronidazole and intravenous levofloxacin was initiated. So even before they knew it was Clostridium, they assumed it was Clostridium, and they began on the Clostridium antibiotics, is basically what they're saying. A stool immunoassay for toxin A was negative, but the toxin was detected by culture and cytotoxicity assay. So they filtered the stool and they exposed the filtrate. So if you take the stool and you filter it, like any of the toxins or anything that's secreted should be in the filtrate. So they exposed the filtrate to cell culture, and they showed that the toxin was actually there. Therapy was changed to oral vancomycin, which is better than the levofloxacin, and the patient responded with resolution of diarrhea and abdominal pain. This is an example of severe C. diff disease after antibiotic exposure in an immune-compromised patient. Remember, all of your transplant patients are immune-compromised because they have to take immunosuppressive therapy. With a characteristic presentation of the pseudomembranous colitis. The diagnostic problems with immune assays are well known and have been replaced by PCR that target the toxin gene. Treatment with metronidazole is preferred, although vancomycin is an acceptable alternative. We don't see the same levels of resistance to metronidazole in other types of bacteria like we do to vancomycin. So vancomycin to treat C. diff will work, but it potentially exposes other bacteria like Enterococcus to vancomycin, which can lead to the development of resistance to vancomycin in other members of the gut microbiome. So vancomycin-resistant Enterococcus, you might remember from a couple weeks ago, is a big problem. So if we have an antibiotic where we don't see the development of resistance all that easily, that's probably the one we would prefer. And so C. diff is considered to be one of the highest threat levels with the CDC. It can cause life-threatening diarrhea. Infections occur most often in people who've taken antibiotics for other conditions. It is the most common healthcare-associated infection. And again, this is from 2017, 250,000 cases in hospitalized patients. These are patients who were hospitalized for something else. 250,000 of them developed this infection, and almost 13,000 died. So you literally went to the hospital for something else and you died of diarrhea. And about $1 billion in healthcare costs. So it's a big deal. Okay, questions about Clostridium difficile? Okay, so the next species we'll talk about is Clostridium perfringens. And this is one that causes what they call gas gangrene. Basically what that means is, so this is an anaerobic organism. So it causes infection in the deep tissues. And what can happen is it produces gas as a byproduct of its metabolism. And so you can actually, if you have a patient with this, if you like press where the infection is, you can hear the bubbles and you can feel the bubbles under the skin of gas. So it causes soft tissue infections, so gas formation in the soft tissue. Weirdly enough, that's apparently not painful. But It can also get into the muscles. And in the suppurative myositis, you're getting the production of pus. That's what that word means. Suppurative is you have pus. But you're not having muscle necrosis. And then you can get muscle necrosis, right? Myonecrosis. So the bacterium can lead to death of the muscles, right? Those are some of the deepest tissues you have, right? They're underneath your fat. They're right along your bone. They're pretty deep in there. And so it's a painful, rapid destruction of the muscle tissue, systemic spread with high mortality. So if you catch it before it causes all that, you're probably okay. But when it starts to become necrotic, that's an issue. It can also cause GI symptoms. It's, I don't want to say a common cause of food poisoning, but it is a cause of food poisoning. Self-limited food poisoning, watery diarrhea, no fever, no nausea or vomiting, short duration, self-limited. In some patients, though, it can cause a necrotizing infection in the GI tract, especially in the jejunum. And of course, this would cause pain. It causes vomiting, bloody diarrhea, and it causes an infection in the peritoneal area. So it gets out of the intestines and can cause an infection in the abdominal cavity. It has a lot of different toxins. There are a number of different types or strains of Clostridium perfringens. All of the types have toxins that can cause hemolysis, bleeding, and tissue destruction. And I won't ask you to remember all of the different toxin types. I'm just showing them to you because they're what cause a lot of the disease manifestation. The beta toxin is more commonly associated with the strains that cause GI illness because they can cause intestinal stasis, right? Normally your intestines move through peristalsis, and that's what brings like fecal material out and moves food through the digestive tract, so it can stop that. It can cause necrotic lesions. There's the epsilon, which can also cause the increased permeability of the GI wall. That can help the bacteria get out, but it can also allow fluid in that can cause diarrhea. There's an iota toxin that also causes necrotic activity and vascular permeability. And then they have this enterotoxin that they often, the strains that produce it as they're transitioning into the spore. It causes altered membrane permeability, so it can cause diarrhea. And it also can serve as a super antigen to overstimulate the immune system. So again, I'm not going to ask you to remember all of the different types of toxins. Like there's not going to be a question, list the 5 toxins that Clostridium perfringens can produce. Like, no. But it has a lot of toxins that basically make it a very effective pathogen. So type A is the one that we would see, like the strain, I guess you'd call it type A. It's found in the GI tract and in nature. It does form spores. B through E don't really survive in the soil, and they can sometimes colonize the GI tract. They don't form spores as readily. That's why they don't do well in the soil. Again, it causes a variety of different disease, and type A is just the most common type of Clostridium perfringens that we see. And so what we worry about, this is a picture showing like the necrosis that you can see. So it's another one of those organisms that can cause necrotizing fasciitis, right? The tissue destruction. So remember, and this isn't super common in students of your background, but especially in people who don't have a background in microbiology, they think like necrotizing fasciitis is a bacterium or is an infection. Necrotizing fasciitis is the disease that can be caused by a number of different kinds of bacteria. So this is one that can do it. And of course, it can cause that muscle destruction. The myonecrosis. So you have extensive bleeding, extensive loss of red blood cells, a lot of hemolytic activity. Food poisoning is fairly common. It's mostly an intoxication. So it's not like you've ingested the bacterium. You've most likely ingested the toxin itself. Short incubation, cramps and diarrhea, under 24 hours. And if you reheat your food appropriately, you're probably going to be just fine because that destroys the toxin. But the necrotizing enteritis of the jejunum, it's pretty rare. It's generally associated with one of the less common strains of this bacterium, but it has a high mortality rate, and weirdly enough, it's associated with pork and sweet potatoes. I don't like pork, but I love sweet potatoes. Just cook your food. Cook your food well. So this is an outbreak of Clostridium perfringens associated with corned beef. So if this was spring semester, I guess we'd worry a little bit more. During St. Patrick's Day. I've actually never eaten corned beef. On March 18th, the day after St. Patrick's Day, in Cleveland, they received telephone calls from 15 people who became ill after eating corned beef purchased from one delicatessen. After publicizing the outbreak, 156 people called the health department with a similar history. In addition to diarrhea, 88% complained of abdominal cramps, 13% had vomiting, which developed an average of 12 hours after eating the implicated meat. An investigation revealed the deli had purchased 1,400 pounds of raw, salt-cured meat, and beginning on March 12th, portions of the corned beef were boiled for 3 hours, allowed to cool at room temperature, and then refrigerated. On the 16th and 17th, the meat was removed from the refrigerator, heated to 48, and served. 48 is not that hot. Cultures of the meat yielded greater than 10,000 colonies of Clostridium perfringens per gram. The health department recommended that if the meat could not be served immediately after cooking, it should be rapidly cooled in ice and refrigerated. Before it's served, it should be warmed to at least 74 to destroy the heat-sensitive toxin. Okay, so again, it's one of those, a lot of these food poisonings do go unreported. Like, I don't know, if I got diarrhea, my thought wouldn't be, I should probably call the health department and tell them I have diarrhea. And just sit at home for a day and not do anything. And that's what most people do, but for whatever reason, 15 people decided to call the health department. But 156 didn't until they read about it in the news or heard about it on the TV. So a lot of these, which is scary when you think about like how much you might go out to eat, but a lot of these things just kind of go under the radar. And you can see how things like this can get missed. So they're rapidly growing. So this idea of letting it cool for so long allows the bacteria to grow. For an anaerobe, it's relatively easy to culture. Some anaerobes are tricky. This one's not too bad. You can find it via microscopy and immune assays. And this particular strain is not always an obligate anaerobe. It can be more of like it can tolerate oxygen, aerotolerant. So it can grow sometimes in conditions of oxygen, which is why we can see it in exposed food products like that salt-cured beef. So it's not always a strict anaerobe like difficile, like botulinum, and like tetani that we'll talk about next week. It can be diagnosed through immune assays. Treatment usually begins, if you're going to treat it, before diagnosis due to the rapid disease course. Again, if somebody turns up with diarrhea and that's it, no fever, no vomiting, you kind of just let it go. And within 24 hours, if they're not better, then maybe you would do something about it. But if you suspect the skin and soft tissue infection, if you suspect the necrotizing enterocolitis, that's when you're really going to want to start prompt treatment. For patients who have the skin and soft tissue infections, you want to do debridement of the wound. So you have to remove all of the necrotic tissue to expose the healthy living surface. So for patients who have wounds or who have burns that need to have debridement, this is one of the most painful things that they go through, because you expose all of that like healthy tissue and the nerves and everything, because you have to get rid of all the dead stuff. And high-dose penicillin therapy. Anywhere from 40 to 100% mortality when you have one of those more severe, complicated forms. Okay, so we'll go upstairs and look at our stuff. Monday, we have an exam covering through here. Wednesday and Friday of next week, right after our exam, we don't have class. I will post some videos on some more bacteria, the equivalent of 2 classes worth of videos that you can watch. And we will pick up either with kind of the tail end of the bacteria or the very beginning of the viruses the Monday after. Okay, so we'll go upstairs and record our